Carbohydrates, digestion. We will now review carbohydrate digestion, the process of a carbohydrate-containing food item moving and breaking down from the mouth to the stomach to the small intestine. Once cleaved to individual monosaccharide units, absorption occurs and the monosaccharides are transported into circulation. Digestion involves the mechanical and chemical breakdown of foods. For carbohydrates specifically, the process cleaves or hydrolyzes glycosidic bonds joining monosaccharide and disaccharide units. While we consume carbohydrates primarily in the form of disaccharides and polysaccharides, the end products of carbohydrate digestion are monosaccharides such as glucose, fructose, and galactose, which are then absorbed into the bloodstream. The process of carbohydrate digestion of starch involves the mouth, the stomach, and small intestine. First, beginning in the mouth, we have salivary amylase, which breaks down the alpha-1,4 glycosidic bonds joining glucose units of the polysaccharides amylose and amylopectin. At this point, amylose and amylopectin are broken down to smaller units, including glucose, maltose, and dextrins, which are longer chains of glucose. It is important to note that alpha amylase only cleaves the alpha-1,4 glycosidic bonds joining glucose molecules and does not hydrolyze the beta-1,4 glycosidic bonds of lactose, the alpha-1,2 glycosidic bonds of sucrose, or the alpha-1,6 glycosidic bonds of amylopectin. You will see in this diagram the activity of salivary amylase cleaving the alpha-1,4 glycosidic bonds of amylose and amylopectin. So you see here amylose, which is a starch. It's a straight chain of glucose units, and you see the activity of the alpha amylase breaking it down into smaller dextrins. And then we see the other starch, amylopectin. You see chains of glucose units, and you also see the branching characteristic of amylopectin. We know from previously that the glucose units are joined by alpha-1,4 glycosidic bonds. However, these branches are joined by alpha-1,6 glycosidic bonds. The activity of alpha amylase on amylopectin also breaks it down into the shorter glucose units called dextrins. Next, we see in the stomach there is no further digestion of amylose or amylopectin, as the amylase enzyme is deactivated by the acidic gastric juice. Then we see that the smaller units of maltose, glucose, and dextrins then move into the small intestine. At this point, the pancreas releases pancreatic amylase, further hydrolyzing the alpha-1,4 glycosidic bonds of the amylose and the amylopectin. You can see the dextrins from being broken down from amylose with further activity of pancreatic alpha amylase. We see that broken down to the smaller unit maltose, the disaccharide. And then we see the dextrins from amylopectin with further pancreatic alpha amylase activity have breakdown also to the disaccharide maltose, but also the maltotriose and limit dextrins, with the limit dextrins being the glucose chains being bound by the alpha-1,6 glycosidic bonds. Lastly, as the smaller units reach the surface of the villi and the microvilli of the small intestine, specific enzymes targeting digestion of disaccharides, such as the disaccharide maltose and also isomaltose, become active. So you can see here that maltase enzyme is being used to cleave maltose, the disaccharide, into individual glucose units. And this is from breakdown of amylose. And then we have the further breakdown of amylopectin. We also have maltose being further cleaved by the maltase enzyme to the individual glucose units. And then we have isomaltose, or the limit dextrins, which are going to be cleaved by both maltase but also the isomaltase, with the isomaltase cleaving the alpha-1,6 glycosidic bonds. So then we have the end products of digestion of starch being purely the monosaccharide glucose. Therefore, we see this digestion of the disaccharides occurring primarily at the microvilli of the intestinal mucosa cells. We saw the activity for starch with the amylose and the amylopectin breakdown with the maltase enzyme primarily, but also the isomaltase with the maltase cleaving the alpha-1,4 glycosidic bonds and the isomaltase cleaving the alpha-1,6 glycosidic bonds. But at the surface of the villi and the microvilli of the small intestine, we also see secretion of lactase, which cleaves the lactose, the beta-1,4 glycosidic bonds of lactose. And then we also see activity of sucrase, cleaving the alpha-1,2 glycosidic bonds of sucrose. Here's a great summary diagram providing an overview of each of the carbohydrate digestive enzymes. So we see the enzymes here. We see the glycosidic bonds that they hydrolyze, the compounds they digest, and the site from which they are secreted. And we also see the end products of the digestion process. So it may be helpful in your studying to review this summary slide and each of its components.